So this lecture is going to cover sexual reproduction and meiosis. Why do organisms have sexual reproduction? Why is it a very important thing in biology? Why is it an important thing for life? And then the processes that allow sex cells, things like sperm and eggs, to arise, and why that process is a little different than, say, body cell division and things like that, mitosis. So, sexual reproduction occurs in two main ways. These are fairly easy to tell apart. Both of them have their distinct reasons for being. So, one of them is asexual reproduction. The prefix a means without, so without sex. So, it is asexual reproduction. So, reproduction without sex. So, there is no genetic variation going to occur here. This organism right here, you can see this little amoeba. He's going to undergo asexual reproduction. He's going to literally make two identical copies of himself, split himself in half to end up with two identical clones from the original organism. So these guys have both the same genetics as the original organism here. This is really good if the environment is stable. If this guy's getting enough food, if he has enough nutrients, everything over here is good. He's warm, his temperature is happy, everything in this environment is good. There's not chemicals or anything in the environment that's going to kill him. If he's happy, chances are both of his identical offspring, his identical clones, will also be happy. But if something over here is bad or a disease comes in, everyone is going to be impacted the same. If he's going to get the disease, both of his identical offspring will get the disease. There is no chance that one of them might be immune. They're going to all be identical. So having an identical population is slightly risky, but it allows for this guy to not have to worry about finding a mate, and he can very quickly reproduce, and so this one can very quickly reproduce, and then they can build a population very quickly. If you don't need to find a mate, there's really no purpose in it. It's very expensive to have a baby. So energetically speaking, asexual reproduction is very beneficial for an organism to do, but the trade-off of that is if the environment goes bad or something bad happens, you're probably all going to die at once. Your entire population is very risky, very, very at risk. So, once again, asexual reproduction, one organism is going to replicate its DNA and make two identical copies. So, one copy of DNA is going to be made into two copies. One copy goes to each new cell. Virtually identical copies, clones of each other. There is no difference between these two cells and no difference between the original cell that they came from. Good when the environment is stable. Not so good when the environment is not. So, sexual reproduction, as we do, and a lot of and other animals that you're probably familiar with do as well, is a little more costly, energetically speaking. In fact, it's a lot more costly. You have to grow these little organisms inside of you. You have to provide for them when they're born. You have to feed them. Very, very, very expensive from an energy standpoint. It's also risky for the mother. She has to risk her life to grow her little babies as well. So very, very, very energy costly. But the trade-off of that is if a disease comes in and gets the mother, one of her babies, if not more of them, are probably going to have some slight immunity. Or if the environment goes bad for the mother, it's too hot outside or too cold, one of her babies might be a long-haired cat or a short-haired cat that's a little more apt to survive in the environment than the mother. So all the babies are going to be slightly different than their parents. So that's going to cause genetic change, genetic variation, which could lead to a better adapted baby. Maybe he blends in a little better into his environment than the mother does. Maybe he can run a little faster than his mother. So all of those genetic variations lead to organisms that are different looking than their parents. You can see this little baby up here doesn't look like either of his parents. He's kind of a hybrid in the middle. But he might be slightly taller than his dad, slightly shorter than his mom. But his mom might be not able to run so fast, but his dad might, so he might get his dad's running ability. So he's better able to survive out in the wild by running faster, and he's a little taller. So he gets everything in the best of both worlds. Or he could get the worst of both worlds and be really short and run really and not be able to run at all, and not be able to survive in the wild. So it's just kind of a shakeup of genetics. But you have a better chance of getting something beneficial than being an identical clone of your original parent. So, why is sexual reproduction so common in organisms? Why doesn't everything just make a copy of itself? This is exactly that concept I just went over. You get a chance to shake up your genetics to increase the likelihood of one of your babies surviving. And if I'm freezing to death, and my partner's freezing to death, maybe, just maybe, one of my little baby kitties here will have enough hair and enough fur to be able to survive the cold winter, and our species can continue. Whereas over here, this little amoeba, if a chemical comes in that's going to kill this one, it's going to kill every single one of his offspring because they're all the same. They're all going to be impacted the same. Whereas over here with our kitties that sexually reproduce, he has a little slightly better chance. This one little offspring might be more long hair, might be able to survive the cold just a little better than his mother, which allows their species to survive, whereas all of the amoeba that asexually reproduce died. So sexual reproduction is beneficial because it shakes up genetics. So let's talk about how sexual reproduction works, and what type of cells sexual reproduction occurs in, and kind of what's going on inside of that. Well, the first thing that we need to talk about is the concept of haploid and diploid cells. So every single cell in your body that's a non-sex cell, and sex cells refer to gametes, things like sperm and eggs. Those are the two sex cells are going to carry two sets of identical chromosomes. You can see that in here. One here and one here. They're numbered. 1, 2, 3, 4. Humans have 23 pairs of chromosomes for 46 total. So they're numbered 1, 2, 3, 4, 5, 6. You get the idea. With 2 sets of 1 sets of 2. You get the idea here. You get number 1 from your mother and number 1 from your father. 1 from his sperm, 1 from your mother's egg. So on and so forth for each chromosome. Every single cell in your body is going to have this same setup inside of it. These cells are called diploid. These are going to be normal single cells that you could pull off of your arm, cells that you'll pull out of your liver, cells that you'll pull out of your toenail, cells that come out of your eyeball. Every single cell in your body except for sperm and eggs. Normal single cells in your body. If you could pull one out and look at the DNA, this is what you would see inside. These types of cells are called autosomes. These chromosomes that are found in your normal cells minus the X and Y's, the sex chromosomes, chromosomes number 23, are going to be called autosomes. They're going to control the normal day-to-day operations of your body and how your body looks. Your eye color, your hair color, your skin color, how fast you can run, how tall you are, and things like that. Whereas the sex chromosomes, those guys are not called autosomes, and they're going to be what determines your sex, your gender. Now, every single person on the planet is going to share the same, every single cell and every single person on the planet is going to share the same numbers 1 through 22, these autosomes. These are going to be found in every single person. The only difference is going to be the sex chromosomes, numbers 23. Every person on the planet is going to have these autosomes right here, as well as some sort of combination of X's and Y's. And that's going to be what determines your biological gender. So if you end up with one X chromosome from your mother's egg, you will be biologically determined as a female. The second chromosome that you get from your father's sperm determines what you will be when you are born. So everybody defaults to one X from your mother's egg. That's what she gives you. She does not have a Y. That's the determination for females, is 2 X's. She cannot give a Y, so she automatically gives you an X. So the gender is determined by the father's sperm. So if the father gives another X in his sperm, he has the ability to do that. Males are XY. You're going to end up with a female. If the male sends over a Y in his sperm, you're going to end up with a male. And that's what's going to determine the gender of the baby, the offspring. So the numbers 1 through 22 are going to be determined as autosomes. Those are the same in every single cell. Every single person on the planet is going to share those. The difference is going to be numbers 23, the sex chromosomes. You can see here that all of these chromosomes are identical. They share the exact same size. They look the same. You can even see the colors are the same on some of them. And we'll talk about what the colors difference are in just a little bit. They're the same size, the same shape for the most part. They're just kind of bent a little different, but if they were straightened out, they would be the same shape. Anyway, the reason why they're the same shape and the same size is because these are copies of the same chromosome. This is the one from your mother, and this is the one from your father. They are equal pairs. The only ones that are different are going to be males. If you have an X and a Y, the two X's are going to be identical as for a female. If you're a male, you're going to have one X and then a smaller Y. And that's going to be the only difference in the size of the chromosomes. So the reason why these guys look alike is because they're termed homologous pairs, which means that they are two identical pairs of chromosomes from the mother and then an identical pair from your father. So when cells replicate, they're going to replicate their chromosomes. We'll talk about how this works in just a second. But what you have here is a chromosome. And this contains one chromosome from your mother, and then its replicated copy over here. One chromosome from your father right here, and then its replicated copy over there. And those two things together are known as homologous pairs. So because they're the copy, homologous means the same. I'll post a video about all this stuff later to give you guys a little more elaboration on this as well. So homologous pairs, homologous meaning the same, pairs of chromosomes. So they are identical. This is chromosome number, maybe let's say 1 from your mother. This is chromosomes 1 copy from your mother that will be given to its copied cell. Chromosome 1 from your father, and then chromosome 1's copy of that. So chromosome 1 is going to carry the same genes from your dad as the same genes from your mom. And that's what makes these homologous. This is the gene here, let's just say, that codes for eye color. Well, your dad has eyes, so he needs the same chromosome, so he obviously is going to have a copy of that. It just might be slightly different than your mother's, but it's going to be in the same chromosome and the same place. This is the instructions on how to make a human. They need to be in the same order in the same place to make a human successfully. And this is why they're identical copies. So, even though they're identical copies, the information that's inside of them could be slightly different. And these are called alleles, these genes. This is a gene, essentially. And these are, like I said, this is the gene that codes for, let's say, eye color. Well, your father might have blue eyes and your mother might have green eyes. So obviously the gene for eye color is there because they have color inside of their eyes, but the version of that gene is slightly different. And those are called alleles, those different versions of the same gene. Hair color, skin color, height, they're all the same gene because we all have those things, but the versions of them are slightly different, that allele. And that's why things like hair color, skin color, eye color are different. They're variations across people because of the different variations of those alleles. So, like I mentioned, every single cell in your body, except for sperm and egg, is going to be considered a diploid cell. It's going to have 2 copies of every single chromosome inside of your body that does what it needs to do to carry out its cellular day-to-day business. It's going to reproduce itself and make a copy that's identical to it. Sperm and eggs, on the other hand, they are going to be termed haploid cells, which means they only have one set of chromosomes inside of them. This is the sperm here, and this is the egg that it's penetrating into underneath it. So why are they like this? Why is haploid and diploid a thing in sperm cells and not our body cells? So your sperm here is going to be carrying half of your father's genetic information. The egg is going to contain half of your mother's genetic information. Well, this is going to have 23 chromosomes inside of it, and this is going to have 23 chromosomes inside of it. If we made an identical copy of the sperm, of the body cell, into a sperm, the 46 cell chromosome containing body cell just makes a copy into a sperm that contains 46 chromosomes. And the egg does the same thing. You're going to have 46 chromosomes inside of an egg. If they unite together, you're going to have way more chromosomes than you're supposed to, and the byproduct won't be a human. It probably won't even survive. So what needs to happen is the sperm and egg needs to have the numbers of chromosomes halved. Split in half from the father, split in half from the mother. So when these organisms, sperm and eggs, unite, you have the correct amount of chromosomes to create a new organism that's identical to what it's supposed to be, that will survive, that has the correct number of chromosomes. So that haploid, half the number, sperm, that haploid, half the egg, they come together, they unite to form one diploid cell called a zygote. One diploid, which has both copies of the chromosomes from mom and dad, which is what we're looking for. One diploid cell called a zygote, and that's the very first cell of a developing offspring. So once that offspring occurs, you can see here the cells In the father, the sperm and the cells in the mother, the eggs are going to be produced by a process called meiosis, which we'll talk about in just a second. So once meiosis occurs, the sperm and the eggs are generated, and they unite. These haploid sperm and eggs are going to unite to form a diploid zygote. That zygote will then divide by a different process, not meiosis, called mitosis, recreating identical copies of diploid cells. So haploid cells over here, diploid cells over here. Your body needs to have diploid cells. Everything in your body is diploid except for sperm and eggs. So eyeball cells, arm cells, everything else. So we're just going to make a bunch of different types of diploid cells to create a fully developed human. And then once that fully developed human reaches puberty, if you're a male, you'll start to produce haploid sperm. And if you're a female, your eggs are produced in utero. You're born with all of the eggs that you will need. They are produced, or that you will have, I should say, and they are produced by meiosis in utero before you're born. So, the formation of meiosis, by meiosis, that makes sperm and eggs. These are called germ cells. The sperm and eggs. And I mentioned they divide by that different process called meiosis. So let's go ahead and talk about meiosis in depth, how these sperm and egg cells are made. So every cell in your body, including the cells that make these, are diploid. So how do we get a diploid cell that has 2 copies of every single chromosome into a haploid cell that has 1 copy? That's kind of difficult, kind of an interesting process. So let's go ahead and talk about meiosis in detail, how we do that. So essentially, what's going to happen, if you're familiar with the process of mitosis, this is mitosis twice in a row with a couple of extra steps inside of it. So you start out with this diploid cell. That diploid cell is going to essentially divide 2 times without duplicating its DNA the very first or the second time. So kind of an interesting little concept. So we'll kind of take an in-depth and look into this. Like I mentioned, I'll also post a video that will go over this in detail, as well as kind of how this works in kind of a video form, so it's a little easier to understand. So you can see over here, we have diploid. We can see the green arrows. These cells are diploid. Diploid, 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 and then we can see the switch to haploid as well, when half the numbers of chromosomes. So you can see diploid, chromosome 1, chromosome 1, chromosome 1, 2, 2. You can see them both there, identical copies. They will be split in half, and then split in half again. So diploid body cell up here, going to be used to create sperm and egg. Now these are germ cells, a little different process, but it's kind of neither here nor there. So essentially what's going to happen is we're going to split these little X's in half until we get one side of that little X into a cell by itself. And this will either become sperm or an egg. So here's how this works. DNA is going to replicate once, but the nucleus divides twice. So the very first stage of cellular division in meiosis is called meiosis 1. There are 1, 2, 3, 4, 5 stages of meiosis 1. They're fairly easy to understand. It's essentially mitosis twice in a row. So the very first thing that's going to happen inside of this is we take our diploid cell that exists inside of your body. You can see the video, our little pictures down here as well. So the diploid cell inside of your body is going to replicate its DNA. It's already done. Everything that's needed to make cell division occur has also been produced. The centrioles, centromeres, and centrosomes and things like that are already produced. The DNA has been condensed down into chromosomes. So, are going to be ready, I should say. So, prophase 1. Your chromosomes are going to condense, become visible inside of the nucleus. So, chromosome 1 is going to be paired up with chromosome number 1 as well from its identical copy. So, chromosome 1 here has been replicated, so now we have chromosome 1 from mom, and then identical copy of chromosome 1 from mom, so it's like a little X. And then chromosome 1 from dad is paired up with its copy, so another little X. So 2 X's right next to each other, chromosome 1 from mom and chromosome 1 from dad. They're right next to each other, 2 little X's. So the next thing that's going to start to occur in prophase 2 is the little spindle fibers. These are essentially a fishing reel, and these little centrosomes here kind of worked as the fishing rod. They're embedded inside of the cell membrane of the cell. Little tiny spindles, these are microtubules, are going to start to form. This is the fishing line. It will eventually attach to the middle of the chromosomes, and the little centromeres will reel them back in and separate them apart inside of the cell. What's a little later. So the first thing that's going to occur in prophase 1 is those spindle fibers are going to start to occur, and the nuclear envelope is going to break down. Those chromosomes have already formed up with their partners, and they've already linked up the two X's together. So chromosome 1's paired up with chromosome 1 from mom, and they're linked up and ready to go. So the nucleus is going to start to break down the nuclear envelope, so the spindle fibers can get inside of there to start pulling the chromosomes apart, because we need to get this 2-copied cell into 1-copy cell, and that's the whole point here. So the next phase is metaphase. Metaphase means middle. So what's going to happen here is the nuclear membrane is totally gone. The chromosomes, you can see chromosome 1 here from mom lined up with its pair. Chromosome 1 here from dad lined up with its replicated pair. Chromosome 2 from mom lined up with its replicated pair. Chromosome 2 from dad lined up with its replicated pair. Fairly easy on that one. They're going to line up in the middle with their partners. So one's going to line up right next to copy of 1 from dad. Copy of 1 from mom's going to line right up to copy of 1 from dad, or 2 from dad, I should say. 2 from mom, 2 from dad, 1 from mom, 1 from dad. They line up right next to their copy along the middle of the cell, something called the metaphase plate. And you can see that right down here. They're all lined up in the middle of the cell. This is a diploid cell. Keep that in mind. It's got 2 copies of each chromosome inside of it. These spindle fibers are going to extend fully and extend and then attach to the centromere in the middle of that chromosome. And once again, this is the fishing reel, and this is the fishing line. The fishing line is now going to stick a little fishing hook inside of the centromere of that chromosome. So the next phase is called anaphase 1. Ana means apart. So the centromeres, the chromosomes in this point, are going to be pulled back apart. The cell is going to start to elongate a little bit. You can see that. The centromeres are going to start reeling in the chromosomes. This is going to separate them apart. So chromosome 1 from mom and its copy is going to be separated from chromosome 1 and its copy from dad. So they're going to be separated from the middle. They're no longer connected anymore. So 1X is going to go over to one side from chromosome 1, and the other copy of chromosome 1 and its copy from mom is going to go to the other side. And then chromosome 1 from dad and its copy is going to go over here. And then chromosome 1 from mom and its copy is going to go back over here. So this is going to start to separate, taking 1 full X, 2 copies of chromosome 1 from mom, 2 copies of chromosome 1 from dad, separating them apart on to the sides of the chromosomes, or the sides of the cell. And then what's going to happen is we're essentially going to have something called kind of an intertelekinesis, so intertelophase. It depends on the book. This book calls it telophase 1. It just depends on the interkinesis. It depends on the book. But essentially what's going to happen is this cell is kind of going to pause its cell division for a second. What we have is a cell over here that is getting ready to divide again. This cell will divide once and create 2 cells. And this cell has a copy of chromosome 1 from dad and a copy of chromosome 2 from mom. A copy of chromosome 1 from dad and a copy of chromosome 2 from mom. You can see here they're slightly different. The blue one's over here, the red one's over here, and they have their copy with them as well. So these are going to be split again to make a haploid cell. Now, one of the parts I left off on here that I'm going to come back to is something called crossing over. And this is a very, very, very important part of meiosis that occurs in prophase 1. So you can see here, these cells, this little thing here, the chromosomes have now switched sides. This red one is switched with the blue. This is a process called crossing over. Cells that are chromosomes that are homologous, the homologous chromosome pair, they're identical. They can swap sides when they get close enough. Genes can switch. Whole chromosomes can switch. Whole parts of chromosomes, the entire side can switch. Little pieces. It really just depends. And this is also to increase genetic variability. And we don't want an identical copy of dad, so we get mostly dad with a little bit of mom in there. Or maybe this chromosome contains the everything from dad's eye color except for dad's hair color. So a little bit of different thing, get mom's hair color. So it shakes things up a little bit. This is an extra process to shake up genetic variations, crossing over here. That's where those chromosomes, they switch places. So the next thing that's going to occur is we're going to enter meiosis 2. So we're going to end our process here with that interkinesis, where we start with a haploid cell that contains 1 copy of chromosome 1 with its identical copy, and then 1 copy of chromosome 2 with its identical copy. Whereas over here, our original cell contained 2 copies of chromosome 1 with its identical copies as well. So now we have one cell, or one copy with chromosome 1. So those cells are going to divide again. So essentially the exact same thing is going to occur again. Our nuclear envelope is going to break down. Our spindle fibers are going to start to form. They're going to form and reach across the metaphase 2. They're going to bond to the centromeres in the middle. The fishing reel is going to reel the X's back across, except this time the X's themselves are going to separate. The chromosomes separate into their individual chromatids and are pulled to the separate sides of the poles of the cell. The cells then divide in half again, and you end up with haploid cells. So 4 identical copies that have half the number of chromosomes that the original cell did. So each time meiosis occurs, you end up with 4 haploid cells from one diploid cell. And you can see the process that occurs down here. These are the actual dividing cells. You can see what's occurring, how this works. So when this process occurs to generate sperm, you end up with 4 fully functional sperm cells from each meiotic division. When this process creates eggs, you end up with 1 egg cell and 3 non-functional cells called polar bodies. And the reason for this is eggs are very, very, very expensive to make. They need a lot of energy to power building a baby. So they need to be really big, and they need a lot of mitochondria and stuff inside of them. So when this occurs, this division occurs, you end up with 1 cell that's really big, that gets all the mitochondria and all of the good stuff, and then you end up with 3 little cells that don't get anything, and they're just there because they have to be, because that's how meiosis and cellular division works. They don't have anything in them except for DNA. They're lucky if they get a mitochondria, and they break down and are reabsorbed back into the body. So 4 sperm cells, 1 egg cell per meiosis division. So once again, that meiosis, that crossing over concept that I mentioned in prophase 1, is going to increase variability. And there's another thing that I forgot to mention that I'll talk about now called independent assortment that occurs in metaphase 1. So we'll go over both of those a little bit in depth now. So crossing over, what happens? This happens in prophase 1. I pointed out where the colors changed, where the chromosomes switch sides. So we have here chromosome 1 from mom, and its identical copy. It's copy that's being ready to be split in half. Chromosome 1 from dad, and its identical copy. And you can see here that they're really close to each other. The genes are the same. The alleles may be different. You can see the capital A versus the lowercase a. But what can happen is when they get close to one another, since they're so similar, parts of the chromosomes can swap. And when this swaps, you end up with a brand new copy. So instead of having big capitals all the way down, you have 2 little lower cases, and then the rest are capitals. And then you end up with 2 big capitals, and then little lower case letters for this chromosome. And then when it divides and ends up with a different, ends up in a sperm or an egg cell, you end up with something different. So instead of being an identical copy of your dad that's given in a sperm cell, you end up with a slightly different copy of your dad than you had in a sperm cell. So this is an interesting concept to kind of shake up genetics a little bit more. Independent assortment occurs because chromosomes don't necessarily line up the same way or divide the same way every single time. Chromosome 1 could end up on top the first time and from your dad, and then the second time that another cell lines up, it could end up on the bottom. And then the next time the cells line up, it could be in a different order. Every single time the cells line up, they could be in a totally different order. This chromosomes line up. They don't have to be in the same order. You can see it switches every single time. Something's different. And because it's different every single time, the outcome of the independent cells in the end, the sperm cells or the egg cells, is going to be different as well. If they lined up identically every single time, dad's cells all on the top, dad's chromosomes, mom's chromosomes all on the bottom, every single copy would be identical to it every single time. But when you have difference, you can see here this is a different version. We've swapped the blue and the red. You end up with different sperm and egg cells. Just do it again here. You swap the middle ones, different sperm and eggs. You swap them again, different sperm and eggs. You end up with different types of genetic information. So this is another way to help shake up genetic variability. So this is to help ensure that your offspring are different than you, that they probably will be more likely to survive in the environment than you are. This occurs during metaphase 1. So how is it that all these things occur in our body to shake up genetic variability, that identical twins, can still occur? So, when this occurs in humans and every other animal that produces identical twins, essentially what's going to happen is you're going to have a haploid sperm and a haploid egg that unite to form a diploid zygote. That diploid zygote is going to split itself in half, creating two identical zygotes. Since embryo 1, zygote 1, is identical to zygote 2, both of the copies of chromosomes that are found in each of those zygotes are identical to one another. That means they're going to be the same. So as those embryos mature, the zygotes start to divide, the embryos grow, the baby eventually is born. All the genetic information that's contained within them is identical to one another. They are the exact same because they came from a split of the original zygote. That zygote split in half to create two identical zygotes that developed independently of one another. And that's an identical twin. So how does it work for fraternal twins? Well, this occurs when a female releases two eggs at the same time. Identical twins occur when one egg splits into two, which explains the same DNA, this is very similar looks, everything like that as adults. Whereas fraternal twins, twins that don't look the same, can be different. This even explains why they can be different gendered. You can have one male sperm. Fraternal twins, they look different. This occurs when a female releases two eggs at the same time that happen to be, or more than one egg, I should say. You could have 5 or 6 babies at the same time. It happens every once in a while. Fraternal twins occur when more than one egg is released at the same time that's fertilized separately by independent sperm. And this is a really good way to show genetic variability, crossing over, independent assortment, and things like that. Because if every single sperm and every single egg that your mother produced and every single sperm that your father produced were exactly the same, both of these children would be the exact same gender, they would look the same, they would be identical copies of one another, nothing would change. But since they're not, you can see that things are different. This is obviously a male, this is obviously a female. This came from two different sperm. They have different heights. They're slightly different as they look, the way they'll turn out, their heights, intelligent levels, all kinds of things can be different. And that's all going to be determined by the independent assortment of those chromosomes and the random roll of the dice that occur, which one they get and what they get from pairs of their chromosomes from their mother and father when the zygotes are formed from that uniting of that haploid sperm and that haploid egg to form a diploid zygote. So two separate zygotes, more than one, different DNA from different sperm, different eggs, resulting in two totally separate individuals that have separate DNA that look different as well, whereas identical comes from one zygote that splits into two. So, mitosis and meiosis. Meiosis is essentially mitosis 2 times. Mitosis, as a diploid, creates a diploid copy of itself, identical copy. One cell identically copies itself, diploid to diploid. Meiosis occurs in 2 different stages and is essentially going to be making one diploid cell into 4 haploid cells. So that's the difference. Sometimes during meiosis, you can have some problems that occur. You can have something called a non-disjunction, and this occurs when cells have their chromosomes fail to separate. So sometimes the fishing line inside of our little cells don't correctly attach as they should to the centromeres, and the fishing line breaks, or the reel doesn't reel it in correctly. And what happens is you end up with a chromosome that fails to break apart from its partner. And you can see here this one was bound, chromosome 1 from mom, bound to chromosome 1 from dad, copies. They fail to separate. So what happens is when that occurs, you end up with an extra copy of that particular chromosome in the zygotes. You can see over here you should end up with 1 copy of each, or 2 copies. Whereas in this one you ended up with 1 copy in 2 and 3 copies in 1. And this is something called non-disjunction. You can also have it where it occurs in a different step in meiosis. It really just depends on if it's in meiosis 1 or meiosis 2. And then you can end up with 3 in one and 2 in the other, and then 1 in one. It really just depends on which phase of meiosis it occurs. When this occurs, you end up with extra sets of chromosomes, this non-disjunction concept, the chromosomes fail to separate, you end up with an extra set of that chromosome in the end product. And when the sperm is added in, instead of having one copy that you should from the egg and then one copy from the sperm, you have 4 copies overall. And that can lead to some serious problems. So if you have missing chromosomes, this zygote is missing a red chromosome. So is this one. This one has an extra copy. If you're missing one, you can have some birth defects. If you have extra, you can have birth defects. It really just depends on which chromosome it is. There we have 24 to choose from, and it depends on how much or how much extra or how much you're missing of that particular chromosome as to how severe that disease is. Sometimes you can survive missing or having extra chromosomes, and sometimes you can't. It really just depends on the amount or individual chromosome in question. So you can see down here, this is chromosome number 21. This is Down syndrome. And this is a normal karyotype, a person that had successfully occurring meiosis. Their chromosomes split separately. Chromosome 21 is the questionable chromosome in here. And this person has, you can see it's a male. We have one X and one Y chromosome over here. And they have 2 copies of chromosome 21. Now, if you zoom in on someone with Down syndrome, they have 3 copies of chromosome 21. You can see it in their karyotype here. They have an extra copy there, and you can see that visibly expressed if on a karyotype of someone with Down syndrome. That chromosome failed to separate somewhere in the meiotic division, and then you ended up with an egg or a sperm cell that carried 1 extra copy of chromosome 21 than it should have. And that's how you end up with Down syndrome. The more or less of that particular chromosome that's copied, if you have more, you have a more severe case of Down syndrome. The less of that chromosome you have, the less severe the case of Down syndrome is. So if you sometimes they can occur in non-autosomal chromosomes. They can occur in sex cells too. You can have non-disjunction of sex cells. You can have extra X's or extra Y's added. Or sometimes you can have a Y that gets failed to be added at all, a second chromosome. You end up with a sperm cell that's lacking an extra a non-autosome, a sex chromosome. You can end up with just a 1X and something else, nothing, so just 1X. These are a couple of the diseases that can occur down there, the conditions, I should say. That can occur by having extra chromosomes or too many chromosomes or a lack of that second chromosome. And some of the things that can be associated with that. And that's it, guys, for the meiosis PowerPoint. If you have any questions at all about this one, please feel free to email me, and I will get back with you shortly. Have a good rest of the day.